So every cell in your body has just about the same genetic information, but the reason you get hair and skin and eyes and nose is because of epigenetic regulation of that. And so epigenetics really brings together cell fate and disease. And it's actually been an interesting field of study because of a couple of observations that were made a long time ago. One is called the Sweden over Calyx study, and in this is a great example of how environment can actually affect future generations. So in the Sweden over Calyx study, whoops, on that animation, I went the wrong way. What they showed is that there was a period in this village in Sweden where they kept records for generations and generations, in which there was the parents and grandparents had a period of diminished nutrition, and so they had slow growth because they had lack of nutrition in this northern Swedish village. If you then looked 1, 2, and 3 generations later, there was still an effect on cardiovascular and diabetic mortality as a result of the mothers in particular, because women have a limited number of eggs. In a period of nutrition, the epigenetic regulation of those eggs became disrupted, and that showed up in future generations in cardiovascular and diabetic mortality. So that's one study, fine. And, one study may or may not be true, but it was not an experiment, but almost the same thing happened right after World War II, in which there was a massive Dutch famine in 1944 and 1945, and essentially, between the Germans and the French and the US and the British and whoever else kept marching back and forth across the Netherlands, all of their farmland ended up being just completely devastated. And so there was this period of massive famine in 1944, 1945. And exactly the same thing happened. In this case, there were decreased birth weights in the infants as a result of their mothers going through a period of famine even before they were pregnant. So it had a long-lasting effect. So environment and cell fate are really intimately linked, and suggest it's a good idea to eat healthy, I guess. But we're going to focus on a class approach. We're going to focus on how epigenetics affects cancer, and particularly on a phenomenon called tumor suppressors. So tumor suppressors are proteins that dampen down some of those hallmarks of cancer that I talked about earlier. So a tumor suppressor, for example, would regulate apoptosis or regulate angiogenesis. And now you get in a double negative. If you suppress a suppressor, then you're going to upregulate the ability of the tumor to grow. And that's one way that the particular enzyme that we're going to talk about affects cellular growth. In addition, one of the things we've learned, actually earlier this year, is that another important mechanism is disturbing these regulatory genes that affect cell cycle. So there are enzymes that control cell cycle. They're called regulatory proteins. They are very creatively called cyclins because they affect cell cycle. We're not like if any of you are in the Drosophila world, Drosophila scientists come up with the most creative names. Drug discovery people just, it's cycle, so it's cycling. It transfers methyl groups, so it's a methyltransferase. We don't have these exotic names that the Drosophila world does. And those are two things that we've importantly learned, in fact, this year. So if this works, I am going to show you a little video.
Human tissues are composed of cells, and each cell in the human body carries the same genetic blueprint, stored in the nucleus in the form of DNA. DNA wraps around nuclear proteins called histones to form structures called nucleosomes that resemble beads on a string. Histone proteins can be modified at precise locations by a process called methylation. Methylation, performed by a group of enzymes called histone methyltransferases, or HMTs, can change nucleosome structure and make specific genes more or less accessible to RNA polymerases. Genes that are more accessible will be transcribed into messenger RNA and expressed, while those that are inaccessible will not. Histone methylation is a highly regulated process since appropriate control of gene expression is crucial to normal cell growth and division. In some cancers, specific HMTs are overexpressed or genetically altered in ways that cause them to be more active than normal. This leads to unusually high levels of histone methylation and altered gene expression. In this example, the result is abnormal activation of genes that promote cell proliferation, causing the cell to divide rapidly, leading to cancer. Small molecule inhibitors can bind selectively to the altered methyltransferase and can thereby block the activity of the aberrant enzyme, restore normal methylation patterns, and reverse abnormal gene expression. This leads to cancer cell death with minimal effect on normal tissue cells.
So that little video is actually taken off our website. We put it together because we had lots of people asking us what we did, and now we can just send them to our website. It makes it a lot easier, and we've actually gotten a lot of people call us and tell us how much they enjoy seeing that, particularly non-science people, because it's easy to follow along. And later today, I'm going to talk a little bit about careers in science if you don't want to work in the lab, basically. If you love science, but what else are you going to do? That video is put together by it's a small company. It's 4 or 5 people in the Boston area, all of whom are science majors, all of whom were also art majors, all of whom decided that the way they wanted to exercise their love of science was by making scientific videos like that. And they're a terrific group of people to work with. But that's fundamentally what we're going to talk about. We're going to talk about the fact that HMTs can be genetically altered. When they are genetically altered, they become oncogenic. That leads to misregulated gene expression, and that misregulated gene expression can show up as disease. And in our particular case, cancer. It can also show up as metabolic disease. There's a increasingly large body of data that suggests autism spectrum disorders could be misregulated epigenetic phenomenon. Schizophrenia, subsets of schizophrenia are almost certainly an epigenetic phenomenon. Subsets of diabetes, particularly type 2 diabetes, can be an epigenetic regulation. So there's lots of places in which misregulated gene expression can show up as disease. We've chosen to focus on cancer, really for 2 reasons. Cancer tissues from humans are easily accessible by biopsy, so you can do the genetic sequencing. It's much harder to biopsy a piece of your brain and understand if you've got a change in that part of your brain. And secondly, because this is a relatively new area of science, much of the stuff that I'm telling you, or the stuff that I'll show you, has only been discovered in the last 5 or 6 years. And so combining those 2 things, you can gain access to the tissue and we're learning a lot of new science, makes cancer an important place for us to start, and it's a place where there's still significant clinical need. So, repeat what we've been saying. HMTs are control gene expression by placing methyl marks on these proteins called histones, and when they're altered, they become oncogenic. So one of the things that our company actually discovered when we got started 6 years ago is that these family of histone methyltransferases are a large family. There's 96 of these histone methyltransferases within the human genome. And the way we discovered that is we took the sequence, the genetic sequence of the active site of one of these enzymes called the set domain. So within these enzymes is a domain that actually moves the methyl group from the methyl donor to the methyl acceptor. And that enzymatic domain, that protein domain within these enzymes is called the set domain. So we took the sequence of the set domain, and then in collaboration with another small company in the Boston area that was primarily made up of mathematicians that fell in love with biology, we asked the question, how many proteins within the human genome contain this set domain? And when we asked that question, we came up with 96 of them. When you then align those sequences and you say how closely are those sequences to each other, you get what is called a tree. So this is an HMTome tree. The closer two of these, and each one of these branches represents a different protein, histone methyltransferase. The closer they are together, the closer their the closer their sequence is. As it turns out, the closer their enzymatic activity is as well, because the sequence reflects the activity. Of those 96, they're about evenly split, almost exactly 50-50, between a family of methyltransferases that move methyl groups onto an amino acid residue called lysine. So lysine contains an amino group. Amino groups can chemically accept up to 3 methyl groups. So you actually have 4 different forms of methylated lysine. You have unmethylated, monodi, and trimethylated. Likewise, the amino acid arginine, which contains an imino group, can also exist in 4 different forms. A unmethylated form, of course, a monomethylated, and then 2 chemically distinct forms of dimethylated on the imino group. In one form of the dimethylated, both of those methyl groups are on the same nitrogen atom. In the other form, they're on 2 different nitrogen atoms, but chemically distinguishable from each other, not chemically interconvertible, very stable states. They're called asymmetric and symmetric methylation of the imino group. And so these enzymes split pretty evenly between lysines and arginines, and they're very pristine in their activity. If you methylate lysine, you never methylate arginine. And if you methylate a particular lysine within the proteins, you don't methylate another lysine within another protein. They're extraordinarily specific. This is sort of and this was an epizyme discovery, by the way, that there were 96 of these, that they were split between them. This branch over here is kind of an interesting branch because in order to tie these proteins together with these proteins by sequence analysis, you can see this branch comes off in between them. We had to include this branch of proteins. This was proteins of sequence that were of unknown activity. So they were sequenced within the human genome, but nobody had yet figured out what the function of that protein sequence was. Through this genetic analysis, we had to include that branch, that family branch, in order to get this one right here, that one right there connected with that one over there, those sort of at 1 o'clock and 10 o'clock. We had to include this whole branch. So then our scientists then cloned, expressed, and purified one of these and showed for the first time that, in fact, it was a protein methyltransferase. And now, by extension, about half of those have been shown to be protein methyltransferases.
I have to ask, the new ones, are they found in any lower mammals or going down into lower eukaryotes, or are those purely a human construct?
No, they are found in lower eukaryotes. It's really an interesting branch, so not to get too off the track.
Yeah, they're used
in
to
the
it.
in the lower eukaryotes, and actually this branch right here are actually RNA methyltransferases. So they move methyl groups onto RNA rather than onto proteins, honestly, that's an area of science that is just evolving right now. And the function of methylated RNAs is just being elucidated. So that's how they exist. Each of these, while there's 96 in the human genome, lower eukaryotes, any organism that contains chromatin has methyltransferases in them. So, for example, the protein we're going to talk about in a few minutes, we call EZH2. The E stands for enhancer, the Z stands for zest, and zest is a particular Drosophila phenotype. So this protein exists in the fruit fly Drosophila, and the H stands for homolog because it's found in humans. So it's an enhancer of zest homolog, representing the Drosophila nomenclature. And like I said earlier, human people aren't really as creative. So if you look at these HNTs, the next question we asked is how many of these have a genetic alteration within them that appear to be a driver of cancer? And so now in a little bigger where you can actually see it, each one of these green dots is a protein within this HMT family in which there are genetic alterations within that protein that are implicated as a driver of cancer. So in cancer cells, there's actually two forms of mutations. There are what are called driver mutations. Those are the ones that are in control and are really driving, hence the name, the cancer cell to grow abnormally. When you get a cell that's dividing rapidly and quickly, it can just pick up random mutations as it's dividing. Those mutations are called passenger mutations because they're just along for the ride. They have no idea where they're headed, but their cell's growing and they're happy. Those are passenger mutations. Driver mutations are the ones that are causing the cancer. And so when we and this is actually our chief scientific officer, Bob Copeland, who has published a lot of this stuff. So when you look within these, there's about 20 of these that we've identified as having genetic alterations within them to drive cancers. And this comes directly from the human genome sequencing activity because cancers now cancer cells are now sequenced in their totality, completely and often. And so you can ask a question in the cancer genome database, is there an alteration with an HMT or not? And you can ask that bioinformatically using mathematical algorithms that have been developed to analyze those databases in sort of big data science ways. And so when we did that, we came up with at least this set of proteins being genetically altered in HMTs. That NSUN2 is the one that we actually showed for the first time as a protein methyltransferase. So what kind of cancers can they cause? Well, again, this is the same set, and now here's the cancers that have been associated with those types of, with alterations within those enzymes. So as your eye looks across there are blood-borne cancers like myeloma, like non-Hodgkin lymphoma, that one up there, that's EZH2, acute leukemias like that other red one over there. There's subsets of breast cancer in which there are genetic alterations, so PRMB14, WISC1L1, NSUN2, HARM1 over here. Has breast cancer, there's SMID3 does too, but sort of 11 o'clock. That has alterations in genetically defined subsets of breast cancer. And by now you've probably looked across there. Pancreatic cancer, lung cancer, prostate cancer. In every case, in these cancers, we have found a subset of patients that have a genetic alteration within the relevant HMT, and that appears to be the driver of that cancer based on cell culture work. So what kinds of genetic alterations then are found in these cancer cells that have alterations within the HMT? And we've seen virtually every kind of genetic alteration that you can imagine. We've seen chromosomal translocations. So in the chromosomal translocation, where the T stands for translocation, and then chromosome 8 and chromosome 11 are associated, in which a chunk of chromosome 8 has gotten moved to chromosome 11, and that's what a translocation means. So chromosome translocation between chromosomes X and 18 affect EZH2. Translocation at chromosome 11 in the Q portion of chromosome 11 at position 23 is subject to a lot of translocations, and so there it's just 11q23, and then there's about almost 60 different other chromosomal locations that get translocated in there. That causes a form of leukemia. 511 is acute an AML, another form of leukemia, myeloma, diffuse large B cell. You can see in every case the genetic alteration, translocations, in this case point mutations, or complete deletions. Each of those then are drivers of cancers. Each of them affect a particular histone methyltransferase, and each of them have a different effect as a result of that chromosomal translocation. So in the case of EZH2, which shows up twice, because of the chromosomal translocation, it shows up actually 3 times. In the case of X18 translocation, which causes a form of sarcoma. Sarcoma is a soft tissue disease, occurs often in kidney and lung and muscle, soft tissues. and in the synovium, the lining of those soft tissues. Altered methylation of a particular residue, K27, so remember K stands for lysine in the single letter abbreviations. K27 means position, the lysine that's in position 27 of a protein called histone H3. So H3K27 refers, it's sort of like an area code, if you will. H3 is the protein, K27 is the particular location within the protein. Altered methylation there. There's a point mutation within EZH2 that leads to altered methylation. It causes diffuse large B-cell lymphoma or follicular lymphoma. And there's another deletion, there's a deletion of chromosome 22q that causes a form of tumor called malignant rhabdoid tumor, another form of tumor called atypical rhabdoid tumor. That's of particular interest to us at Epizyme. It's a pediatric cancer for which there is no effective therapy. These kids typically present with tumors at 2 years of age. Current therapy is surgery, radiation, and high-dose chemotherapy. And so you can imagine giving a 2 to 5 year old radiation, chemotherapy, and surgery. There's significantly treatment-related effects. And the kids are almost all die by the time they're 6. There's a few, the survival rate is about 15 to 20%, and the ones that get past 6 years of age are all dead by the time they're 10 years of age. So it's a particularly horrible form of leukemia. And we have an inhibitor of EZH2, and so we're pretty excited about the fact that we are very, very soon going to start trials in these kids with MRT with an EZH2 inhibitor. So with that, I'm going to transition a little bit to EZH2 and spend the last 15 minutes or so talking now very specifically about one particular enzyme that is in this family of HMTs. It's genetically altered, and how we understand that that's causing, in this case, non-Hodgkin lymphoma. But what I'm going to say is also true for the kids that have the malignant rhabdoid tumor or the ATRT. So just to stop for a minute. We have now talked about how important mathematical algorithms are to discover genetically altered cancers, how important it is to understand the medical need of what we're doing, the medicine for drug discovery. We're now going to transition a little bit to some chemistry, some cell biology, and those types of activities within drug discovery. And we're going to start by saying, what's the need? Because drug discovery is really tedious and long and hard and so much fun at the same time. But it could take you 10 years to from beginning to end or more to discover a drug. One of my sons is 28 now, or something like that, 27 or 28. We started working on a drug when he was 4 years old, and when he was in high school, we got FDA Food and Drug Administration approval to sell that drug. So for his entire life, he was hearing about this drug. For basically 14 years, he was hearing about the development of this drug. So these drugs become as close to you as your children because it takes a long time to do it. And so if it takes so much time, you may as well be certain that there's some medical need for what you're going to do before you get started down this 10 or 15 year journey. And so in diffuse large B-cell, it's actually a cancer in which there's a drug called rituximab that has improved outcomes. So about 60% of diffuse large B-cell lymphomas are currently cured with rituximab combined with 4 other drugs. That's what R-CHOP means. So it's a cocktail of 5 drugs that's led to about a 60% cure rate. Current approach to this, if you're not cured by R-CHOP, is to get increased doses of chemotherapeutics. That's what salvage chemoimmunotherapy means, with additional antibodies, the immuno part, followed by something called ASCT, which stands for allogenic stem cell transplant. So stem cells are taken out of your, either from a donor, typically out of your own marrow in this case, because it's allogenic. those stem cells are then grown up and then they're re-injected back into you, and that helps to correct the underlying defect. But if you fail R-CHOP, or you fail allogeneic stem cell transplant, you really have no options left. Survival rates, if you fail R-CHOP, are 5-year survivals are less than 20%. If you fail ASCT, 5-year survivals are 0. So first line therapy, R-CHOP, really effective. If you fail R-CHOP, really no options. And that's what we're focusing on. So in the world of drug discovery, everything you do is regulated by government. In the US, that's the Food and Drug Administration. It's different names in different countries. And so everything you do has to be approved by the Food and Drug Administration. So the components of CHOP, this cocktail of 4 drugs, are approved by the regulatory bodies for NHL. Rituximab is the only agent specifically approved for diffuse large B-cell lymphoma. And while there's lots of things that are under development, there's still this massive clinical need for agents for people with diffuse large B-cell lymphoma that fail first-line therapy. And so that's what we're focused on. It's worth doing, in other words. And this just shows you some of those survival curves. So if you like epidemiology, if you really like to study large populations, this is the kind of data that comes out of epidemiological studies. So this is PFS, means progression-free survival over years with 2 different drug treatments. You can see the survival curves here. So 75% of the patients die by 5 years. In this study, it's about 75% at 10 years. You can see if you fail after R-CHOP, it's similarly dire, 80 to 90, 10 to 25% survival rates. And so that's the therapy that we're going towards. Similarly, in a form of lymphoma called follicular lymphoma, it's got a similar kind of analysis. And this is the kind of analysis we do before we start a project. Is it an area in which there's been progress? Is it an area of unmet need? Do we have a regulatory path forward? Because everything we do is controlled by the Food and Drug Administration. If we can't figure out how to get their agreement up front, we probably won't start the program. You have to have regulatory approval, and you need to know what your competitors are. You need to know if you can beat the agents that are out there or not. And so in every program, we start this kind of activity before we even initiate our program. So how big are these populations? This on the left here shows the incidence of non-Hodgkin lymphoma, particularly a form of NHL called B-cell lymphoma. So incident is how many new patients arrive each year. And there's about 155,000 patients that get B-cell non-Hodgkin lymphoma. And B-cell NHL actually splits into 2 big populations, approximately 50-50. This top one, there actually, over the last 2 years, have been astonishing progress made in agents to treat what's called activated B-cell lymphoma. And that is not a cured disease, but it is not a disease that has the need of the lower half here, germinal center non-Hodgkin lymphoma. So there's about 72,000 of these, new cases of these every year, that split up into various subpopulations. If you look at prevalence, because you can live more than a year, that's new cases per year. If you live 2 years, obviously there's more people with it. That's what prevalence is. There's about 113,000 patients currently with the form of non-Hodgkin lymphoma that we're interested in. And so that sort of gives you a feel for how big the disease is. It's a bad disease. There's a couple of 100,000 people a year that have this disease, and that's the population that we're going after. And now we have to switch and explain a little bit of anatomy. So we've talked about other things, but right now I have to orient you on the anatomy. So this is a lymph node. You have afferent lymphatic vessels that are bringing the lymph into the lymph node. And when those afferents come in, they enter what's called a germinal center, which are these little things. So basically, B cells, particularly form of white cells called B cells, are produced in the bone. That's why they're B. They are immature. They're not quite sure what they want to do when they grow up. They get put out into the lymph. They come into the lymph node and they go to the germinal center. When they go to the germinal center, that's what they decide what they want to be when they grow up. And that germinal center is an anatomically defined space, as shown there in that H&E stain. But it's a very important nursery for these immature B cells. Because what happens when they go in there, when that naive immature B cell goes into the germinal center, it becomes a germinal center B cell. I told you we're not very creative in our names. And then it diverges into one of multiple pathways, actually. If you follow this line that goes sort of towards 2 o'clock, it becomes what's called a memory B cell and plays a role in fighting infections, or it can become a plasma cell, again, playing an important role in fighting infections. But what these blue boxes show is the number of cancers that happen when a B cell at some particular point in its maturation gets confused and has a genetic alteration that causes it to grow abnormally. And so you can get chronic lymphoblastic leukemia, you can get a variety of lymphomas, classic Hodgkin's lymphoma, mantle cell lymphoma, what else? Multiple myeloma, diffuse large B cell, particular subtype. All kinds of leukemias result from these B cells having a genetic alteration at some point in their life and then growing abnormally. And we were over the last, this is actually knowledge that's come out over the last 3 years and has only just been published from our labs and from a lab of a scientist at Weill Cornell, which is the medical school in New York City. When the naive B cell goes into the germinal center, it starts off with low levels of this protein EZH2 and low levels of BCL6. When it enters into that germinal center, it then massively upregulates EZH2 and BCL6. And it needs to do that in order to mature, to move from an immature B cell to a mature B cell. If it decides to head the other way on that branch, it actually continues to hold EZH2 down. So upregulation of EZH2 becomes a switch on this pathway where the B cell is headed. And if it abnormally upregulates EZH2, BCL6, and then another protein that isn't expressed up here, BCL2, what happens is that massive upregulation of these three proteins, and particularly EZH2, is sufficient to cause these forms of lymphoma. So that genetic alteration, the upregulation of EZH2 and BCL family members, cause those forms of leukemia that are down in that lower state. And EZH2 appears to be a critical role in all of that because the product of EZH2 activity is methylation of this residue K27 in histone H3. So H3 K27 trimethylation is the product of EZH2 activity. And it's the ratio between di and tri, turns out, is critical. Stuff we just published earlier actually in June. That ratio between di and trimethylated K27 turns out to be the important switch. And if you get too much K27, you develop this oncogenic phenotype. And there's 5 different ways in which you can upregulate that K27 methylation. You can do it with this set domain mutation. The set domain's that domain that transfers methyl groups. A mutation there can lead to too much activity. You can make too much protein, you can overexpress it. EZH2 sits within a complex called PRC2. If you upregulate other members of that complex, then you get too much EZH2 activity because there's other members of the complex that are upregulated. You get loss of function, which is what LOF means, of some other proteins whose job it is to downregulate trimethylation. So if you lose the activity of a protein whose job it is to downregulate trimethylation, you get an upregulation of that. And so we became particularly interested then in the point mutations within EZH2 because, and this is data that we published in Nature Genetics and Proceedings in the National Academy of Sciences a couple of years ago, because a single point mutation within a tyrosine at position 641, so that shows the DNA, the protein sequence of EZH2 with tyr 641, which sits in the catalytic site. a single mutation there in which that tyrosine is changed to phenylalanine, asparagine, histidine, serine, or cysteine can result in excess trimethylation, and that's what these bar graph shows. So this shows wild-type enzymes' ability to methylate, to go from 0 to 1, 1 to 2, or 2 to 3 methyl groups. So it's not very good at putting 3 methyl groups on. If you come over here to these mutant, you see exactly the opposite profile. They're not very good at putting the first one on. In fact, these can't at all. They're okay at putting the second one on, and they're really good at putting the third one on. So the interesting observation we made was that in all of these patients that had this mutation, they were all heterozygotes. They all had one copy of wild-type protein and one copy of the mutant protein. And so what's happening is that the wild-type puts the first one on, then the mutant protein says, oh good, the first one's on, and it proceeds to put the second and the third one on, and that imbalance, and so there's an imbalance between the di and the trimethylation as a result of the heterozygosity of this particular enzyme. And we showed that, and that's summarized on the top there. We showed that both using peptide substrates, which is what's shown here, as well as using cell lines. So these immunoblots show di and trimethylated H3K27. All these other lines are control proteins. And in these 4 cell lines that only have wild type, you see an excess of dimethylation over trimethylation. In the mutant cell lines, you see no dimethylation, all trimethylated. And that's the imbalance that I was talking about as shown on an immunohistochemical stain. And so we made a small molecule inhibitor showing up in the right hand.
Boy, I was waiting the whole time for
There's finally
an
some
organic
chemistry.
compound. Right.
I
You
did
gotta test organickers.
Yeah, you need to name, so the test is, what is this called? What is that called?
Yeah.
That's why you need to take organic, so you can understand what all
Preach
the
it, brother.
So we made that, our chemists made that molecule. It inhibits both the wild type and the mutant enzyme with about a potency of about 2.5 nanomolar Ki. It doesn't affect any other of those 96 HMTs. That's shown in this bottom right here. The size of these dots indicates the Ki, so the bigger the dot, the more potent the compound. So you only see big red dots at EZH2 or the mutant forms of EZH2, and no effect on any of these other HMTs, so more than 20,000-fold selective. It's orally bioavailable. That's important. When you give it to animals, it actually gets absorbed. Most of the time, that's preferred route for taking any medicine. It's not that easy to build into a chemical. As it turns out, your gut, your liver, and your kidney are extraordinarily efficient at keeping foreign chemicals that it's never seen before out of your system, which is a good thing. We have to try and bypass it in order to get our drugs in. So it's orally bioavailable. It only inhibits K27. When you combine it with dexamethasone or prednisone, any of you that have sort of taken a steroid as an anti-inflammatory probably took dex or prednisone. If you combine those relatively safe drugs with 6438, you get a huge regulation of the activity, which is important because we can increase the potency of the compound by understanding the underlying connection between biochemical signaling, and most importantly, we see effects in animal models. So I'm going to go through the next few pretty quickly because that's just data to illustrate what I said already. We get inhibition of the methyl, K27 methyl mark as we increase concentrations, and that occurs over time. You only see an effect on the K27 methyl mark. You don't see effects on any other methyl marks within the cell as measured by antibodies. On the left-hand side are growth curves with time on the x-axis and number of cells on the y-axis. You get time and then concentration in these different colors. Concentration-dependent killing of cells with mutant on the left side, but no effect on cells without the mutant. So we know that this is a driver mutation for these cells. The cells are addicted to this genetic alteration. If you take those cells and you put them in animals and let a tumor grow up till it's about 200 cubic millimeters, and start treating those animals orally twice daily with 3 different concentrations of 6438. I should say all of them, when you're doing drug discovery, because the chemical names are so long, they all get a number, and we only refer to them as the number because nobody can ever remember the chemical names until they get to a point where they get a generic name, and then they get a trade name. So acetaminophen is a generic name, Tylenol is the trade name, and then it's got a long chemical name that nobody ever remembers. Sorry.
Yeah.
The chemists always remember.
I'm really feeling dissed right now. That's okay.
If you stop treating the animals here, the tumor's completely gone, so they've been completely cured. No tumor grows back. And we can see inhibition of that methyl mark in the tumor. You want to be sure you can see it in more than one cell line, so we showed it in multiple cell lines, and that's what this data says. So independent of what else might be going on in the cell, what other passenger mutations are in these cells, they're all dependent upon the driver mutation, EZH2, for their activity. And now I'm going to jump ahead, just because of time, to where we are with this program, 6438. So this program we've shown is effective. I skipped over, we actually shown it's safe in animals. Animals can tolerate extremely high doses of it. So about a year ago, we requested from the Food and Drug Administration approval to go into humans with this chemical. They said we could. So we have what's called a phase 1 study. So that's a study in which a chemical is tested in humans for the first time. That's what phase 1 means. And really the objectives of phase 1 study is to define the maximum tolerated dose, MTD, how much can humans tolerate of this, or what's called a recommended phase 2 dose. So phase 1 is the first entry into human. Phase 2 is when you test your chemical actually in patients for the first time, and you need a dose for that, so that's the recommended phase 2 dose. There's some secondary objectives there that are related to how well is the drug absorbed, pharmacokinetics, and how much effect is it having pharmacodynamics. So we started that cohort. As of the end of August, we had completed 12 patients at a 100 milligram dose, a 200 milligram, and a 400 milligram dose. We sequenced the EZH2 so we know if it's mutant or wild type patients. We also determined what form of lymphoma they had, so we knew that we had NHL patients with a particular form of lymphoma that did or did not have the point mutation. And in those 4 patients, we actually had four of the patients that had a variety of diffuse large B-cell lymphomas. We had 3 out of those 4 patients that showed a response in the disease. And I want to illustrate that with just one of those patients. So this is sort of the medical part of the talk, and we'll finish with this. So this is a 23-year-old male who had a particularly aggressive form of lymphoma called primary mediastinal B-cell lymphoma, PMBCL. ECOG is just a rating on how bad the lymphoma was. He happened to have wild-type EZH2, which is pretty interesting. Here's his clinical history. He was diagnosed in February of 2013, and where it says 27 November, he entered into our clinical trial in November. So between February and November, he had failed 4 different treatments. This one is a combination of 6 drugs. This is a combination of 5 drugs. This is a combination of 4 drugs, getting increasingly toxic chemotherapeutics. Failed all those lines of therapy. In fact, had the disease progressing through those last three lines of therapy. He got 200 milligrams of 6438, which led to a reduction in this K27 mark that we've been talking about. So we know we inhibited the enzyme in a human being. And what's shown over here is what's called a CT scan. So it's an image basically through his abdomen. And where that red circle is, that's the tumor. There shouldn't be a connection there. So he went on in November. By May, he had almost a 75% reduction in that tumor. Now, this is a patient that had failed 4 lines of therapy, each of with between 4 and 6 chemotherapeutics. Got roughly 6 months of 6438 as a single agent, and has a 75% reduction in his tumor volume. More importantly, remember all the way back to the beginning, we talked about one of the hallmarks of cancer being a change in metabolism, a change in ATP uptake. you can actually image that using a radioactive form of glucose called fluorodeoxyglucose, or FDG. And the brighter the red spot, the more FDG is taken up, the more of this radioactive chemical. And so you can assess how fast the tumor is growing by how much glucose it's taking up. Cancer cells grow so fast, they use glucose as their primary source of energy. So between November and May, that red spot has dropped so that it's indistinguishable from the other organs that are in that scan. So that tumor has shrunk, and it's switched its metabolism back to look like a normal tissue, not an abnormal tissue. And that's really why it's worth the 10 years of doing this stuff, and why it's worth trying to integrate chemistry, anatomy, physiology, epidemiology, sequence, anything you can think of, because of this kind of effect. We'll get another readout from this patient shortly, and see where he is. So that's sort of drug discovery. We started with 1 micromolar inhibitors in 2010, and we entered phase 1 a year ago, and a year after that, we were seeing effects in patients. So it's a long road, but it is so much fun and so much gratifying. And I just want to finish by actually recognizing my colleagues at Epizyme, as well as our partner, a Japanese company called Eisai, and more importantly, the patients who volunteer for these trials, taking chemicals that have never been in humans before, but are willing to do that, not only because of the potential for them, but also because of the potential for society. And thanks for the time, and sorry I ran a little late, but hopefully it wasn't too tedious for you.
Thank you.